Hello, my name is Valentina and today I'm going to present TMEM33 as a prognostic biomarker of cervical cancer and its correlation with immune infiltration. But first we need to talk about what is cervical cancer. Cervical cancer is the third principal cause of death in females. It starts when the cervix cells go through change known as a dysplasia, but if those cells aren't destroyed, they may become cancer cells. Cervical cancer can progress from an early stage to a more advanced stage if it's not treated quickly. Another important aspect is immune infiltration. Immune infiltration is linked to disease progression and chronicity. Also, immune cells infiltration participate in the resolution of inflammation, imposing a highly dynamic view of the inherent process. The general objective in this research was evaluate the TMEM33 as a prognostic biomarker of cervical cancer and its correlation with immune infiltration. Methods. We have four methods. The first one was cell lines and cell cultures. So, to study the cervical cancer, there were used 4 different cell lines for cervical cancer and 1 cell lines to represent the human normal epithelial cell lines. So, the human normal epithelial cell lines was the HCAD and the cervical cancer lines was HeLa, CHA, C33A, and Keski. Another method was the cell transfection that consists in transfer siRNAs to cells by using lipofentamine. Quantitative RT-PCR was another method that helped us to detect and measure some pathogens during each cycle of PCR. And the Western blot analysis is a technique used to detect a specific protein using an electrophoresis. So the results. First one, we have the expression of TMEM33 in cervical cancer cell lines. Here we can see the TMEM33 in the 5 different cell lines and the GAPDH that is the control. This control helps us to standardize the results. And here we have the change of TM33 expressing levels. What's the meaning? That in 2 of the 4 cell lines of cervical cancer we have change introducing the TMEM33 with the siRNAs. Like we can see in the HeLa and CHA, here we know that is decreasing the level of cells. This clone helps us to also view the cells because with TMEM33 the cell proliferation decreases as we know here. And with immunofluorescence for cervical cancer cells that basically show us the difference between having the TMEM33 and not having this protein in HeLa and CHA cell lines. For the discussion, we have 3 statements. The first one was said by Shen, Zhao, and Li, that was that TMEM33 may be involved in the progression of metastasis because, as I said, an overexpression of this protein can have a high risk of expressing a cancer. So I agree. The second one is said by Deepak, Bempati, and Naharahu that say that microenvironment can stimulate development of various tumors. I agree. And the third one was said by Tian, Shen, Li, Wang, and Wang that said that immunosuppressive cells can cause an immune escape and TEM can promote metastasis. Because in general, an overexpression of this protein that help us to have a high risk of prognostic of cancer. But if we inhibit this protein, we have less opportunity to have cancer, in this case cervical cancer. For the conclusions, we have that evaluate a disease with different molecular biology techniques could help us to find an appropriate treatment according to the patient, and also to develop more information about cervical cancer and in general other types of cancer. And a conclusion for this article is that TEM33 is an excellent biomarker that could predict the early span of cervical cancer.